So, to understand genetics, you have to go back to the gentleman who pioneered the field. Gregor Mendel is called the father of genetics, that's him. He was an Austrian monk who lived in an abbey with other monks, and they had a communal garden. And he was a trained scientist at the time, but he did some studies on these pea plants and ultimately came to explain the modern laws of genetic inheritance. And even though we now have a much better understanding of genes as a whole than Mendel ever would have had, his general concepts are still shown to be true today, much as they were in the 1800s. So it's pretty cool, because a lot of science from 150 years ago, we've since improved upon to tremendous degrees, but Mendel's basic ideas about genetic inheritance, they're still taught today, as you can see, so it's pretty cool. Genetics, study of heredity. Heredity, transmission of traits from one generation to another. Mendel wanted to explore how organisms passed on their genes. It's not, I ate some peanuts and my throat is dry. That's all it is. So, genetics is a study of heredity, and heredity is the transmission of traits from one generation to another. When Mendel was alive, one of the dominant ideas was the blending hypothesis. This taught that basically mom was red paint, dad was white paint, their child would be pink paint. You are a mishmash of the sperm and egg cell from your parents, ultimately passed on traits from both, but you are just a hodgepodge mix of both of them. The thing that the blending hypothesis did not teach was that genes, when passed from mother and father to the offspring, retained their uniqueness when they came together to make the child. Let's say you have red hair. Your parents don't, your grandparents don't, but your great-grandmother does, or did. Same hair. When she was younger, she had red hair just like you did. So, how did that trait skip a generation, 2 generations actually, and show up in you completely unchanged from what it was 2 generations ago, when your great-grandmother had the same hair color? Blending hypothesis did not explain this. Blending hypothesis would have taught that the red hair color was gone, blended out, and yet it showed up again, unchanged, after skipping generations. This was an insufficient hypothesis for how genetic inheritance worked. Mendel knew that. He wanted to explain how it worked, and he did. Ultimately, through his pea plant experiments. So let's get to those. There is a pea plant flower. Mendel, by the time this was done, he actually he published an article about it. By the time he published his article, he determined that individual organisms pass on distinct, he called them heritable factors. We now know them as genes. And that these genes were passed on, and they did not become a mishmash. They retained their uniqueness when passed from parent to offspring. How does he figure this out without any of the modern equipment that we have? Well, let's talk about how he did. He figured it out by starting with some controlled breeding. So here's some pea plants. You got purple and white flowers. Mendel, first of all, made sure the plants did not self-pollinate. So if you remember from Flowering Plants Lab, which we did, that was the last lab that we did. If you remember that, pollen from one plant needs to enter the carpel and fertilize the ovule of the other plant. Plants are, most of them, monoecious. It means they are both male and female structures for reproduction. And so Mendel cut off the stamens of the purple flowers, used a paintbrush or other equivalent device to transfer pollen from the white flowers to the purple flowers, and thus artificially pollinated them. This guaranteed that he knew which plants were the 2 parental plants. They produced peas. He took the pea seeds, planted them, and take a look at all of the offspring made by that union. All of them are purple, every single one. So, Mendel first needed parental plants, one purple flower, one white flower. Together they made the parental generation, or the pea generation. That's these two up here. Here. All of the offspring produced by that union would be the F1 offspring. And if you look at them all, every single one is purple. Blending hypothesis would have taught us that purple was the dominant physical trait, and the white coloration was blended out after simply one generation. It's gone. It's blended out. If these guys were to pollinate each other and produce seeds, the next generation and the one after that should continue to show purple coloration. The white color is gone. That's what the blending hypothesis would have said. Okay. So, parental generation, purple and white, offspring produced by that, all purple, they're called the F1 generation. This next part harkens back to the previous chapter when we talked about mitosis and meiosis. We talked about cell division. Do you guys remember the word diploid? I hope you do. What it means is 2 copies of each chromosome. If a cell or an organism is diploid, it has 2 copies of each chromosome. In an organism, such as yourself or a pea plant, you have mostly what are called somatic cells. These are all of your cells other than egg cells or sperm cells used for sexual reproduction. Those are not diploid. We'll talk about what they are in a little bit. But all body cells, all somatic cells are diploid, 2 copies of each chromosome. For instance, again, it's not this simple, but just for illustrative purposes, eye color. If there were a single gene that controlled eye color, you have 2 copies of it, one from each parent. Your dad would have given you 1 gene for eye color, and your mom gave you 1, and now you have a pair of that gene. You have 2 copies of it. So, you're diploid. The flowers are diploid too. There's our flowers. Purple one, white one. These X-shaped structures are chromosomes, right? And notice how purple flower has this particular part on the chromosome that codes for a particular gene for flower color. So, on one particular chromosome, you've got 23 pairs of chromosomes. I don't know how many pairs pea plants have, but on one particular pair of them, there's going to be a section that has the gene for flower color. Since the flower itself is diploid, it has 2 copies of that gene for flower color. It's got the copy here, it's got the copy here. Each flower has 2 copies that code of this gene that code for the color of the flower. The white flower has 2, purple flower has 2 as well. Notice how I've used different colors to represent the genes for flower color in the 2 different colors of pea plant. That's because they have different versions of that particular gene. Flower color is a character. A character is a feature, a heritable feature. That means it's passed on from one generation to the next. A heritable feature that varies among individuals. Well, you can't look around you because we're not in a classroom, but if we were in this classroom, we'd all look different. There's obviously a number of physical features that are different among us. Hair color would be one, eye color would be another, height would be another. These are all characters. They're controlled by our genes, and they vary among a population of individuals. In our pea plants, there's a lot of different characters. One such character would be the color of the flowers. There are multiple versions for most genes. think about hair color. There are several different colors, naturally occurring colors of hair. Several versions of that gene, and in this case, hair color, it's a lot of different genes. But for our pea plant flowers, there's only two versions in this case, purple and white. When you're looking at things in genetics, oftentimes if there's only two versions of a gene, an English letter is used to represent the two different versions. In this case, the letter P is used. We use letters when there's two different versions of a gene because it's pretty easy to have two different versions of a letter, capital and lowercase. In pea plants, coloration, purple or white, is called a simple dominance inheritance pattern, which means that there is a dominant version of the gene and a recessive version of the gene. If they occur together, you're going to see the dominant one. And in this example, in Mendel's experiment, purple petals, purple flowers, are the dominant trait. White flowers are the recessive trait. So we use capital P to represent a copy of the gene for being purple, having purple flowers, and a lowercase p to represent the gene for white flowers. So let's go back then. Let's go back a slide. Our purple flower, we said that it has two copies of the gene for flower color. Here's one, here's two. Both of them are colored purple, which means both copies of the gene for flower color for our purple pea plant, say that 10 times fast, would be big P, the purple gene. Our white pea plant has two copies of the lowercase p, the white color allele. You haven't heard that word yet. The white color gene. So we've got purple plant, two copies of the purple gene. White flower, white plant, two copies of the white color gene. Big P, little p, little p. Okay. Mendel used those two plants as his P generation, his parental generation. He mated them together. If you remember how human chromosomal inheritance works, each parent passes on one of their two copies of a particular chromosome. So if these two pea plants were going to reproduce together, one copy of the chromosome will be passed on to the offspring, the other one won't. Same thing for the white flower. And which one gets passed on? Random chance. The offspring then are going to get one of each letter from each parent. From the big parent, or the big P purple parent, sorry. It's going to get a copy of the purple gene. That's all that that parent can give. The white flower, the only gene it can pass on is the lowercase p, which means all of the offspring produced by this union will have one copy of each type of gene. Okay. Move on here. So here's the word that I used a little while ago that we hadn't defined yet. The word is allele. An allele is an alternate version of a gene. So alternative versions of a gene are called alleles. All right? In this example, purple allele and white allele. In Mendel's P generation, the purple flower had 2 purple alleles, while the white flower had 2 white alleles. Big P, little p. Another word to note is the word homozygous, right? Homo means same, so in this case it means same alleles. The purple flower is homozygous dominant because it has the dominant physical trait, the dominant allele. The little p of the white flower is homozygous recessive because it has 2 copies of the same recessive allele. Both homozygous, but homozygous dominant, homozygous recessive. Punnett squares. You've probably made these at some point. Punnett squares show us all possible offspring that can result from the union of 2 parents. And let me look at my phone and make sure, A, that it's still recording, which it is, and it looks like I got more space on the left side. So we're going to make a Punnett square over here. Punnett squares show all possible offspring that can result from the union of 2 parents. Unfortunately, I do not have a purple marker. That's okay. So, rule number 1, I want you to remember this. If there's one English letter, then the Punnett square is going to have 4 total squares, 4 possible offspring. All right? If there's 2 English letters, it gets a little more fuzzy. So we'll get to that later. So 2 parents, you're making a Punnett square, go above and to the left of the Punnett squares themselves. A little bit better. So, these are the possible offspring. To the left and above the columns, that's where you're going to have possible gametes. And I'm illustrating here with sperm cells and egg cells to really hit the point home. The male, or the functioning as a male parent flower, right? We're going to say that that was the white flower. The alleles it had were white and white. I will use blue to represent the purple allele. So our other parent in this union had 2 copies of the purple gene. So parent number 2 can pass on either the first white allele, that's one possible sperm cell, or the second white allele, that's the other possible sperm cell. Parent number 1 can pass on the first purple allele or the second purple allele. Those are possible egg cells. So then you fill it out like a multiplication table. Big P with little p. And I think that's going to happen for all 4 possible offspring. Every single one is going to be big P, little p. That is how Punnett squares work. They show you all the possible offspring produced by the union of 2 particular parents. In Mendel's experiment, he got these offspring, and all of them collectively were called the F1 generation, and they all looked purple because complete dominance is how this works in pea plants. And purple is completely dominant over white coloration. So, all of them are purple. But notice that the white allele was hidden. It was masked. It was still there. You just didn't see it. That's why the blending hypothesis doesn't work. It didn't take into account the fact that these alleles were recessive and hidden for generations. Please note that this is called complete dominance. There's another thing called incomplete dominance when the recessive allele is not completely hidden. We'll get to that later. So homozygous describes both of the parents here having 2 copies of the same allele. Heterozygous is the term that describes all of the F1 offspring because they have 2 different alleles. Mendel decided to pair a couple of his F1 offspring together and see what happened. This would make the F2 generation. Let's go ahead and do that Punnett square up here and see what that makes. Both of the parents would have been heterozygous, big P, little p. So parent number 2 could have passed on a sperm cell with the purple gene, possibly, or it could pass on a sperm cell with the white gene. Same thing with parent number 1 over here. So now let's fill out this Punnett square. Offspring possibility number 1. It gets the purple gene from both parents. It is homozygous dominant and looks purple. Possibility number 2. It gets the purple gene from the male parent and the white gene from the female parent. It's heterozygous, but it still looks purple. Possibility number 3. It gets the white gene from the male parent, the purple gene from the female parent. It's heterozygous and still looks purple. And then possibility number 4, it receives the white gene from both parents and it looks white. That was the real kicker in this experiment for Mendel. It showed up, the white color showed up after skipping a generation, totally unscathed, exactly as it looked in his original P generation. It wasn't blended out, it just was hidden, it was masked. And so that was the major aspect of this that taught Mendel about genes and the uniqueness of genes and how they maintain their uniqueness when passed from one generation to the next. All of these offspring, by the way, would have been Mendel's F2 generation. A couple of terms to note: genotype versus phenotype. Phenotype is an organism's physically expressed trait. Genotype is an organism's actual genetic makeup. And here's a way to really distinguish them. These 2 hypothetical pea plants have the same phenotype, right? They're both purple. However, genetically, they're different. This one's heterozygous, big P, little p. This one's homozygous dominant. Different genotypes, same phenotype. So keep in mind the difference between those. Genotype's the actual genetic makeup. Phenotype is what it physically looks like. Here's another good way to help you understand with difference between phenotype and genotype. If an organism shows the recessive phenotype, if there's a white pea plant, it has to be homozygous recessive in genotype. For instance, in the pea plant example, it's little p, little p. However, if something shows the dominant phenotype, purple flowers, you have no way to know just by looking at it whether it's homozygous dominant or heterozygous. So how can you tell? You can perform what's called a test cross. I'm not sure if you can see the picture too well, but that's a black lab, and that one is a chocolate lab. I doubt you can see the fact that it's brown very well, but that's okay. Let's talk about labs and lab fur color. There are 3 principal varieties of Labrador Retriever fur color, and at least 2 of them, it's a very simple genetic component that causes the fur color. So, in labs, big B, dominant allele, codes for black fur. Lowercase b, recessive allele, codes for brown or chocolate fur. Just like our pea plants, each dog is diploid, which means each dog has 2 copies of the gene for fur color. Yellow labs, it's a whole other gene that causes that. So we're not going to worry about yellow labs. This is the black lab up here. Chocolate lab is down here. If you look at a chocolate lab, you know, based on its phenotype, brown fur, what its genotype has to be. The only way you're going to have brown fur is if you have 2 copies of the recessive gene, the recessive allele, little b, little b. But if you're a black lab, you could be homozygous dominant or heterozygous. So if you've got a pet black lab here, how can you know which of these your dog is? One possible way is to look at the parentage. Right? If the parent of this black lab was a chocolate lab, then you know it had to get one of the brown genes from that parent. But what if you don't know the parentage? If you don't know parentage, one way to do this could be a test cross. If you have a black lab, please don't actually do this. There are enough puppies that need adopting without doing a scientific experiment right now to produce additional puppies. But this is how you could do it, hypothetically. One English letter, the letter B, so it's a 4-square Punnett square. Your black lab, we'll put over here. You're going to want to mate a black lab with a chocolate lab and see what the puppies look like. Let's say that your black lab is homozygous dominant. If you mate that black lab with a chocolate lab, every single puppy will be a black lab, because the only gene that your black lab can pass on is the dominant one. On the other hand, if your black lab is heterozygous, then roughly half of the puppies should be chocolate labs. And in fact, if you get even just one puppy that's a chocolate lab, you know your black lab must be heterozygous. And that's called a test cross. It's a way of testing the dominant phenotype to see which genotype it actually is. All right. This next slide summarizes Mendel's paper that he published regarding patterns of inheritance. So I'm just going to read through these real quick. Number 1, Mendel noted that alternative versions of genes, which we now call alleles, account for variations in inherited characters. And if you hear a strange sound, it's starting to pour outside, so that's what you're hearing. So alternative versions of genes account for variations. Just look around. If we were in the classroom, look around the room, and you'd see a lot of variation. For each character, so for each physical feature that an organism has that's controlled by a gene, they inherit two alleles, one from each parent, because living things generally are diploid. If the two alleles of an inherited pair differ, so heterozygous, then one determines the organism's appearance and is called the dominant allele, the other has no effect and is called the recessive allele. This is true for traits that involve complete dominance. There are some traits where recessive alleles are not totally masked. Mendel's last statement, his last hypothesis, a sperm or egg cell carries only one allele for each inherited character because allele pairs separate from each other during the production of gametes. So again, you've got, this is just hypothetical, it's not actually this simple. You could have two alleles for eye color. You could have one brown, one for blue. You would pass on one of those to your children. And if you have more than one kid, then it's a random chance each time. You might pass on the brown gene for brown eyes to one child and the gene for blue eyes to the other child. Again, eye color is not that simple, but if it was, that's how it would work. Mendel stated this, and this is true for complete dominance. It's not true for all traits. Some traits do not involve complete dominance. In humans, the type of simplicity that we see with our chocolate lab example and our pea plant example, we don't usually see it in humans. Most human traits are controlled by many genes. Good, still recording. So, it's called Mendelian genetics, involves complete dominance in physical traits controlled by single genes. This is not common in humans. But we'll look at a couple of genes in humans that are controlled by one gene, including albinism. This is an albino squirrel. To be albino means to lack all melanin, no pigment. The key here is you look at the eyes. If the eyes are pinkish or reddish, then the organism is albino. There are albino humans, but it takes 2 copies of the gene to show it because being albino is recessive, right? And 1 copy for normal pigmentation, you will show normal skin pigmentation. Sickle cell anemia is caused, or sickle cell trait, I should say. That'd be a better way to put it. Anemia is the disease. Or I should say anemia is a symptom caused by sickle cell trait or sickle cell disease. But sickle cell trait is controlled by a single gene. Cystic fibrosis can be, and achondroplasia can be as well. That's Peter Dinklage. He's probably the most famous actor with achondroplasia. So it's a type of dwarfism, but it's different from these other traits in that it is a dominant allele. So if an individual has 1 copy of each, the recessive is normal height. The dominant allele is achondroplasia dwarfism. So it's unusual for a lot of these single cell traits to have the condition be the dominant allele. Part of the reason why it is for this one is because achondroplasia it's not a harmful condition to have. So, most traits in humans, polygenic. Polygenic means multiple genes. Most human phenotypic characteristics are polygenic. A couple examples, eye color and hair color. So I've used eye color as a simple example here a couple times, but it's not that simple. Eye color is controlled by multiple genes, so is hair color. So is your height, so is your skin color, so is a lot of stuff, most human things. There's a picture of eyes, and here's at least 2 of the genes we've isolated that we know influence eye color. You do not have to memorize any of that. Any of this. So, Mendel looked at complete dominance. There's also incomplete dominance. Incomplete dominance is when one allele does not completely mask the other one. So, an organism that's heterozygous is going to have an intermediate physical characteristic. Snapdragon flower color is a good example of it, but I prefer hypercholesterolemia. Hyper means above, cholesterol means cholesterol, and emia means blood condition. So hypercholesterolemia means high blood cholesterol. High blood cholesterol levels. So, if you've got no copies of the gene, okay, if you have 0 copies of the gene for this cholesterol addition, and actually I wrote that wrong. There you go. There we go. If you have 2 copies of the normal gene, you have normal blood cholesterol. So the big A is normal. So if you have 2 copies of that gene, you have normal blood cholesterol. If you have 2 copies of the gene for elevated cholesterol in the blood, you have very high blood cholesterol levels. And I hope this is all showing up on the screen. I remember that side of the screen had a little bit less coverage. Yep, it is. Awesome. So if you had 2 copies of the recessive gene, you'd have extremely high blood cholesterol levels. But if you've got 1 of each, your blood cholesterol is slightly elevated, potentially enough that you might need certain drugs to lower it, you need to watch what you eat, exercise, etc., but it's manageable. People who have 2 copies of this gene for hypercholesterolemia, doesn't matter what they do, their blood cholesterol is going to be through the roof, and they'll need medication for it. So this is an example of incomplete dominance. Codominance. This is fun. This is blood typing. So we're going to ignore the positive-negative inheritance for this. We're just going to look at the lettering for blood types, A, B, and O. So in terms of the actual blood types, they're the phenotypes. There are 4 of them. A, B, and O. I'm actually going to stop it here because I think I've gotten well past 30 minutes on this thing. And we'll record the other half of this in another video. All right, welcome back everybody. So, continuing our genetics lecture, we are at codominance, when 2 alleles, which are alternative versions of a particular gene, are equally expressed. It is called codominance, and human blood type works like this. The 4 blood phenotypes, ignoring positive and negative, are A, B, AB and O. There are actually only 3 blood genotypes. There is the A genotype, the B genotype, and then the recessive genotype. Both the A and B genotype are dominant. The lowercase I that you see here is recessive. Now remember, each person is diploid. They have 2 copies of each gene. So everybody will have some combination of 2 of those blood genotypes. Let's just put all the possibilities up here. I want to make sure that that is on camera. It is. Perfect. So these are all of the possible genotypes that you could have for blood type. What are they actually talking about, by the way? What does A and B even mean? Well, they refer to antigens that are present in your blood. Type A blood produces type A antigens. Type B blood produces type B antigens. The recessive allele codes for producing no antigens. What's an antigen? It's something that your body would ordinarily recognize as a foreign, potentially dangerous substance, and it would react accordingly. However, if you have the genes that code for the production of a certain type of blood antigen, your body knows that it's supposed to be there and it won't attack it. So type A blood, people with type A blood, they produce type A antigens. Type B blood produce type B antigens. And AB blood produces both. Let's just draw that. The way I'm going to draw these antigens is nowhere near accurate, so please know that. Type A antigens, we're going to represent with this symbol. So, somebody who has type A blood is going to have A antigens with their blood and recognized by their body as a normal thing. Type B antigens we're going to show as a little semicircle. And someone with type B blood would have type B antigens in their blood recognized by their body as a normal thing. People with AB blood have both. Their body recognizes both A and B antigens as being normal. Type O blood is somebody that has no antigen recognition. So if you have type O blood, your body doesn't recognize any of those antigens as being normal. So what if I'm type O? What if somebody with type A blood tried to donate blood to me? Well, my body does not genetically recognize type A antigens as being normal, and so my body would attack that foreign blood, and that reaction would ultimately, most likely, kill me. So I can only receive blood from other people who are type O, because their blood doesn't have any antigens either. On the other hand, if you're type AB blood, you can get blood from anybody, because your body recognizes all the antigens as being normal. How does this relate to genetics? Well, you've got 2 copies of each individual gene. And by the way, Genotype usually refers to an individual. So this thing over here where I said blood genotypes, it's really much better stated as blood alleles, right? Versions of that gene. Genotype is in reference to an individual's combination of alleles. So if you took any notes on that, make sure to fix that. I apologize for that error. So here are the genotypes. Each of them is going to yield a particular blood type. I think you can probably figure out what they are. To be type O, like me, you have to have 2 copies of the recessive gene. To be type A, you simply need 1 copy of the allele that codes for type A antigens. Same story with B. If you've got both A and B, then you are type AB blood. And that's this codominance idea. It means that both A and B alleles are equally and fully expressed. It isn't some intermediate half A, half B thing. It's fully A, fully B, both shown, both expressed. And when they occur together, they're co-dominant alleles. So, blood type, codominance. Thus far, everything we've been looking at has been monohybrid crosses. Mono means 1. A monohybrid cross is going to be something where we're just looking at 1 character. And so there's only usually 1 English letter. Things like this with blood type, where there's multiple dominant alleles, you get these funky things with the capital and lowercase. But generally speaking, monohybrid cross, 1 letter. What if you're looking at two different features? Then what? Then we get a dihybrid cross, in which 2 letters are involved. Mendel, he liked his pea plants, and he did seed color and seed texture, looked at those together as a dihybrid inheritance experiment. So, in pea plants, in Mendel's pea plants, seed color had 2 different alleles, capital Y, lowercase y, with yellow being the dominant color, which I thought was odd because, I mean, peas are usually green, right? But yellow is dominant, and green is recessive. And for seed shape/texture, the dominant trait was being round, and the recessive trait was being wrinkly. I know wrinkly is not really a shape, but it's kind of a texture/shape. So you know what's going on. So those were the two characters that Mendel looked at. So what did he start with? Mendel used homozygous parental plants. Now remember, we're looking at 2 different characters here. So each adult pea plant is going to have 2 copies of the R and 2 copies of the Y. Homozygous, round. Homozygous, yellow. That's the first of Mendel's plants. Yellow and round seeds. The other homozygous possibility, remember homozygous means same alleles, is going to be 2 little r's and 2 little y's, which is going to be green and wrinkled. Mendel bred those 2 together, and they produced offspring. Now you could make a Punnett square for this, but you really don't have to. if you think about it critically. Parent number 1 is going to pass on one R and one Y, right? Because each adult plant has 2 copies of each gene, and they only pass on one of each to their offspring. So parent number 1 could pass on this big R or that big R. It's going to be a capital R for roundness either way. So possible offspring, all going to have a big R from parent number 1. Parent number 2 can pass on either of the 2 little r's. But it's always going to be a little r. And so the offspring have to get a little r from that parent. Same drill with the y's. Uppercase Y from parent number 1, lowercase y from parent number 2. All the offspring are going to be heterozygous for both color and shape. And so the F1 generation will all be big R, big Y, little y, heterozygous, and they're all going to look like the dominant phenotype. Yellow and round, because it's complete dominance in this case as well. If you remember back to the flower experiment, you can probably guess what happened next. Mendel did cross of F1 offspring, and instead of just showing you that picture, I'm going to do what I would have done in class and use Microsoft Paint to go through how this would work for a dihybrid cross. First thing to note, let me make sure, can you see below the screen? Let's see, it looks like I think you can a little bit. 2 English letters yields a 16 square Punnett square. So it's large. Let's get it up here. There it is. Now we just need to make the 16 squares. I know there's a lot of new technology out there, but I still love my Microsoft Paint. I did not make my squares very even, but that's okay, we'll roll with it. So the parents are going to be placed just like they were in the monohybrid cross. One parent goes above, one parent goes to the left. And if you remember, both parents are going to be the same, right? Because they're all going to be F1 offspring, and they're all going to be big R, big Y, little y. So the other parent would go up here. It's going to be exactly the same. Make this a little bit smaller so it's not in the way. But both parents are going to be the same. Big R, big Y, little y. Okay. Each of these spaces to the left and above the columns represent possible gametes, egg or sperm cells, that our parents could pass on. If you remember from our 4-square Punnett square, it was just a single letter that went in those gamete spots. Because if we're looking at just a monohybrid cross with one character, they can only pass on one of each letter. That same general rule applies here, except there's 2 English letters, which means each of these spots should have one R and one Y, because this parent will pass on one copy of each of these pairs. How do you figure out the 4 possible combos of R's and Y's? It's actually a very slick method for it, and you might remember it from an algebra class. It's called the FOIL method. stands for first, outer, inner, last. So let's go ahead and do it with this parent over here on the left. We read from left to right, so we're going with first R and the first Y that you would encounter moving left to right. And so the first possible R-Y combo that could be passed on by this parent would be a big R with a big Y. The next one, the O stands for outer, so the outer R could be paired with the outer Y. That's going to be a big R and a lowercase y. The I stands for inner, inner R with the inner Y. That combination would be a little r and a big Y. The L stands for last. So we're reading from left to right, the rightmost or last R with the last Y, which is lowercase, lowercase. There are our 4 possible gametes. If this was a male flower, that would be, or if it was the male part at least of the flower, these would be sperm cells it could produce. If it was the female part of the flower, these would be egg cells. Parent number 2 up here, you'd want to foil separately most of the time, except we already know that it's the same genetically as the original, or as parent 1 here. So we can just copy the gametes up to the top part. It should be the same. Then what you do is fill it out like you fill out the multiplication table again. Let's do this. Big R, big R, big Y, big Y. I'm not going to read each of these. This is how you would do it. And you'd go onward down the line. When you do the whole thing, you end up with this. So they've got the same combos of R's and Y's that we did. They completed the entire Punnett square. And look what we ended up with in terms of offspring phenotypes. We've got quite a bit of the completely dominant phenotype, yellow and round. We've got 1 out of the 16 possible offspring that's completely recessive, wrinkly green. And then we've got 3 green and round and 3 yellow and wrinkled. That's a combination that did not exist before. That combination did not exist with Mendel's original parental generation. That was the aha moment for Mendel that taught him that each physical gene, actually it's each chromosome, but Mendel wouldn't have known that, but each gene is inherited independently of the other ones. In other words, being yellow and being round weren't passed on to the offspring together. They were inherited separately. And that led to, ultimately, each of these being different from what we started with in terms of combinations. The name for that is, I believe we covered this last chapter, but it's good to revisit it. The law of independent assortment. What it means is that each gene is passed on independently to the offspring. It's not inherited together. Unless they're on the same chromosome, which hopefully you remember from the last chapter. The phenotype ratio was 9:3 to 3:1. Not too worried that you know that number combination, but I want you to know the law of independent assortment. It led to us having F2 offspring. Those are all the offspring in that last picture that are phenotypically distinct, physically distinct, from either of the parental organisms. And again, that's because that law of independent assortment tells us that each paired genes, each gene is inherited separately from the others. We are going to skip linked genes for now. At least the details of it we're going to skip. There's a lot here. Basically, what you need to know from this is that a chromosome is a really long strand of DNA. You have 23 pairs of them. Each pair of chromosomes independently assorts itself, hence the law of independent assortment. On a particular chromosome, there are different genes. You've got thousands of genes, you've only got 46 chromosomes, 23 pairs. So on a chromosome, there are multiple genes. If two genes occur together on the same chromosome, they will be inherited together because chromosomes are passed on together from the parent to the offspring. Hopefully you remember that from the last chapter. So if genes are linked, they occur on the same chromosome and are thus inherited as a pair. Punnett squares, like the dihybrid cross we just did, assume that genes are not linked. It assumes that they're on separate chromosomes. So that's all you got to know about linked genes. All right. Unfortunately, I do not have a karyotype up here. But if you were to look at a karyotype, that's a picture that shows all human chromosomes, all of your chromosomes. You would notice that all of the pairs of chromosomes, they're called homologous chromosomes, if you remember, are the same length because they have the same genes. They might have different versions of them, but they have the same genes. There's only one pair of homologous chromosomes that might be different, and that would be the sex chromosomes. If you're a male, they'll be different lengths. You have 23 pairs of chromosomes, 22 of them do not determine your biological sex. They determine a lot of other stuff, but not your biological sex, and so those are called autosomes. The sex chromosomes are the X and Y chromosomes. If you have two X chromosomes, you are biologically female, and if you have an X and a Y chromosome, you are biologically male. These are passed on from parent to offspring, and it is the biological father who determines the sex of the child. No matter what, mom is going to pass on an X. Dad passes on either an X or a Y. If he passes on an X, it's a girl. If he passes on a Y, it's a boy. So in humans, the father determines the sex of the child. So this is one pair of chromosomes. You have 22 other pairs that do other stuff also. In other species, sex is determined different ways. Birds, for instance, follow something that's similar to humans, but not exactly the same. Birds have the ZW model of inheritance. In birds, it's almost the reverse of what we have in people. ZW is is female, ZZ is male. So, dad doesn't determine the sex of the baby birds. The mom does. So it's similar to humans, but it's actually the reverse in terms of which sex determines the offspring's sex. Honeybees have a strange system called haplodiploidy. What on earth is that? Well, if you know anything about honeybees, you know that there's one primary reproducing female called the queen, and she lays a large number of eggs. Here we go. There's our queen honeybee. She lays a large number of eggs. Some of these eggs will be fertilized by male bees, some will not. We're going to say that the ones with the X in them got fertilized. If they're fertilized by the male, they've just got twice as many chromosomes as they had originally. They're diploid, which means they will develop into females. On the other hand, the unfertilized eggs only have the genes from the queen, one copy of each chromosome, just like a human egg would have one copy of each. If they don't get fertilized, they are males. And so, whether you're diploid or haploid, whether you have one copy of each chromosome or two, determines your sex in honeybees. Some reptiles, alligators are a common example, have sex determined by temperature. Alligators tend to lay eggs in mounds, and the temperature within the mound determines the sex of the developing embryo. I want to say warmer produces males. I really should know this. I'm embarrassed that I don't off the top of my head. But the temperature determines the sex of the baby gators. And there's other reptiles that follow something similar. In humans, the X chromosome codes for a lot more than just your sex. Look at how long that is. There are a lot of genes here. The other genes are called X-linked traits. And these tend to disproportionately affect males. One example is color blindness. So why do guys get disproportionately affected by X-linked traits? Well, let's take a look at it. Color blindness is a recessive trait that occurs on the X chromosome. So an X with a big N is going to signal normal vision, and the X with a little n is the allele for color blindness. Red-green specifically tends to be the one that's linked with the X chromosome. So if you're a male, you have two possibilities here. You could be X big NY, which means you got normal vision, or you could be X little ny, which means you are colorblind. What if you are a female? Three possibilities. Two X big N's, which means that your vision is normal. You could be heterozygous with one big N and one little n, and your vision is still normal because you have the dominant gene. Or X little n, in which case you are colorblind. But notice 2/3 of the possible genotypes have normal vision. In men, it's only half. You only need one copy of the gene for colorblindness if you're a guy to actually show it. The Y chromosome doesn't mask anything. So how is this inherited? Let's say that a man is colorblind. He can pass on either the X chromosome with the colorblindness or the Y to his children. And let's say that mom has normal vision and no colorblind gene. All of the daughters produced in that union would be carriers, but none would be colorblind. But because mom is not colorblind, and she's the one that passes on the X, none of the sons will be colorblind. So, it's an X-linked trait. Color vision in humans is one example of this. Hemophilia is another. Cats, though, I really like this example. Cats, cat fur color in particular, is an X-linked trait. At least orange and black fur are X-linked. So in cats, we've got black fur and orange fur. It's on the same gene that controls the sex, and cats are just like us. XX is female, XY is male. So black fur is the dominant allele, and orange fur is the recessive one. X big B, X little b. A female black cat is going to have 2 copies of the gene for black fur. X big B, X big B. Female black cat. A female orange tabby cat, like the one up there, is going to be X little b, X little b. A male orange tabby is going to be X little by. It's the recessive trait. Just like color blindness in humans, orange fur in cats most commonly is seen in male cats. Because to have orange fur, if you're a male cat, you just need 1 copy of the gene. To have orange fur as a female cat, you need 2 copies of the gene for orange fur. That's why there are a lot more male orange cats than female. To be a male black cat, you need to be X big B Y. What about this female cat? One of each. Well, the cool thing is, cats, female cats with the black and the orange fur gene have random X chromosome inactivation, which leads to patchwork orange and black. Also known as a tortoiseshell cat, or if there's another gene that codes for some white, then you get a calico cat. These are pretty much always female, barring a couple of strange ways that you can get male cats with this. But generally speaking, it is almost always female because you need 2 copies of the X chromosome to show the patchwork black and orange of a tortoiseshell cat. My cat growing up, her name was Whiskers. She was a female tortie, and yeah, you're not gonna have a male tortoiseshell unless it has something called Klinefelters, which is the human term for an XXY individual. There are cats like that. That would be the only way you'll get a male calico or tortoiseshell. Otherwise, they're always female because it's X-linked. So that concludes the second half of the genetics lecture.